Hello, this is Ed Chapman, and this video cast is the final one for photosynthesis. And in this video cast, we're going to focus on something called the Calvin cycle, which is the part of photosynthesis that takes place inside the chloroplasts that absorbs carbon dioxide and uses energy generated by the light reactions, ATP and NADPH, to actually make carbohydrates. So we're going to see how that happens. Okay, the first thing to remember here is that the Calvin cycle takes in carbon dioxide as a reactant, and it outputs carbohydrate. All right, now that carbohydrate is not going to be C6H12O6 glucose directly. It's actually going to be something called 3GP. Okay, and this is a 3-carbon organic molecule that can be quickly assembled into sugars, into carbohydrates. So, for example, if you take two of these, two 3GPs, you can then get 6 carbons, and you can actually build a glucose from that, or a maltose, or a fructose, or starches, or cellulose, or any of the other things you need by assembling these basic carbohydrate building blocks called 3GP. So the output of the Calvin cycle is carbohydrate, but it's a really simple form called 3GP. We'll see that in just a minute. Okay, in order to do this, in order to take carbon dioxide out of the air and change it into a carbohydrate, the Calvin cycle needs a power source. And the power is coming from ATP, all right, which is where it always comes from, just about everything we've talked about. And it's going to need electrons, okay, in order to bond hydrogens into the carbons and the oxygens. You're going to need electrons to do that, to reduce the carbon dioxide. So the electron source is going to be NADPH, which is our electron carrier. And both ATP and NADPH are made by the light reactions, those photosystems we saw in the previous video cast. All right, here is a big scary diagram that's the Calvin cycle, and you guys don't really need to know a whole lot of detail from this. What I want you to see is that it is a cycle. It goes around in a circle. All right. Second thing I want you to see is that it takes in carbon dioxide. Okay, so we're going to take in 3 CO2s are going to go in. All right. Those 3 CO2s are going to go through a series of reactions that eventually is going to output 3GP, which is glyceraldehyde 3-phosphate down here. So this is the output. All right. And so for every turn of the Calvin cycle, all right, you are taking in 3 carbon dioxides, 6 ATPs, 6 NADPHs here, 3 more ATPs over here. Let's see if anything else going in over here. No, not really. So what you get with each turn is 3 CO2s going in and 1 glyceraldehyde 3-phosphate coming out. And these can then be processed into the more familiar energy sources that we've been talking about. Carbohydrates like sugars, even bigger things like fatty acids, and even amino acids. So this is the Calvin cycle is the primary way that plant cells inside of their chloroplasts build monomers, stuff that you need to build all your cell parts out of, from carbon dioxide, and of course water went in the light reactions. Okay, so the Calvin cycle is where the CO2 is going in. Now, another important fact about the Calvin cycle is an enzyme called Rubisco. Now, Rubisco is an enzyme that changes ribulose 5-bisphosphate right here into this 6-molecule material here. So Rubisco is the element that does this change right here. So it's the first thing that actually interacts with the carbon dioxide. So this enzyme is critical because it is the enzyme that actually fixes CO2. And when we talk about CO2 fixation in science, we're talking about the removal of CO2 from the atmosphere and the sequestration or the storing away of it in some type of solid compound, especially in the biomass of living things. And in this case, we're building it into carbohydrates, fatty acids, amino acids, the building blocks of life. All right, now this is what Rubisco looks like. This is a kind of a cool 3D molecule drawing of it. It's a very complicated protein, and it's called Rubisco because it's actually ribulose biphosphate carboxylase. All right, it's the first enzyme in the Calvin cycle, and it's the actual enzyme that catalyzes or the fixation of CO2. All right, and it is the most abundant protein on Earth. So you find Rubisco just about everywhere, and pound for pound, it's the most common protein on our planet because it's basically the basis of the food chain. It's how CO2 is getting into biomass. Now, this is our final slide. It's a big summary slide. This is everything pulled together. We have a cell here, a plant cell. It's got, this is a chloroplast, not a plant cell. And we've got the light reactions over here taking place in the thylakoid. We have it being powered by sunlight. Photosystems 2 and 1 working together to break water down into hydrogen ions and electrons and spit out the leftovers as oxygen. And in the process of energizing these electrons, making ATP and NADPH, which are the products of the light reactions. These then are feeding into the Calvin cycle, which is over here. The Calvin cycle is using ribulose bisphosphate and Rubisco to absorb CO2 from the atmosphere or fix carbon dioxide in the atmosphere to output G3P, which is the basic 3-carbon molecule that's used to build just about everything that cells need to build their parts. Okay? And of course, when the Calvin cycle turns, it's regenerating NADP+, which can go carry more electrons, and ADP, which can then be re-energized. So we got a cycle here of the energy. All right, so this is a really nice slide because it shows you everything that a chloroplast is doing as long as the light is shining, water is available, and CO2 is available. Okay? Thanks for listening, and we have reached the end of photosynthesis. I know you guys are glad to hear that.